This video is designed to introduce the biofiber-based growing medium developed by Bedrock Environmental Services in conjunction with Techly Technical Services, Alberta Innovates Technology Futures, Alberta Agriculture and Rural Development, the National Research Council of Canada, and the Alberta Crop Industry Development Fund. The purpose of this research is to determine the economic and ecological viability of transplanting thousands of plants into a large area in a relatively short amount of time, and to determine which materials would work best. Essentially, we are attempting to grow and install native sod. This may prove to be more efficient than the current method of production and installation, which involves planting each plant individually. Our experience with the current method has shown that it is very costly and time-consuming. Long fibers from flax and hemp plants are extracted in a process called decortication. Plants are cut down, retted, baled, and brought to a processing facility where the short and long fibers are separated from the plant and transformed into a usable form. They pass through a myriad of conveyors and tubing where the plant bits are sliced, diced, and broken down before being deposited into small bales. After the fibers have been separated, they are woven into a 1 foot by 2 foot sheet by TTS, which we have dubbed as a plantable mat. The materials that are currently being tested in the first phase of our research trials are a combination of hemp and wood fibers and flax and wood fibers. The wood fibers are from trembling aspen trees. At the conclusion of these trials, we hope to focus our efforts on one of the materials. The biofiber mats were placed at the bottom of propagation trays and covered over with the growing medium, which consists mainly of peat and potting soil. They were then planted with a variety of native Alberta plants with rooting systems that will test the limits of the biofiber mats. Ideally, the mats would contain the roots for 2 to 3 months, all while maintaining their structure long enough to be planted as one unit. Some examples of the things that we are measuring are root penetration through the biofiber mats, the fragmentation of decomposition of the mats, and plant health. The plants were grown in the trays for 2 months and were given a thorough examination prior to planting. They were planted in 2 separate sites, the first being at the farm in Sangudo and the other in the growing plots in St. Albert. After 3.5 months of growth, we can see that a tray planted with annual wild rye from one of our side experiments has led to the total decomposition of the bio-fiber materials. No traces of the material remain, and the shape of the tray is held together only by the roots of the plants. More information to come as the trials progress.